Great. So I guess I'll do a quick introduction before we get started. So welcome everyone to our fall quarter spree seminar series. It's a great pleasure to have everyone in this virtual room. We wish we could be in person listening to this wonderful talk, but you know, for the obvious reasons, we'll have to do our seminar series virtually this quarter. Professor Balogun and myself, Sinan Keten, we're going to be hosting the seminar series for the fall quarter. Last year, we decided we wanted to do something slightly different with the spree seminars, and we thought it might be nice to organize the seminars into thematic topics per quarter. And the topic we chose for this quarter is broadly related to soft materials, active materials, and topics in that domain. And this theme is going to span quite a few different topics, you know, structural composites, polymeric materials, self-healing materials, architected materials, nanomechanics, and biomaterials. So there's quite a wide variety of subjects, some really in the periphery of civil engineering as we traditionally define it, and some really breaking new ground in this area. So it's a great pleasure to kick off the series with Professor Balogun's talk. He is an expert in the characterization and visualization of elastic waves and phonons in materials. He uses optical experimental techniques for this purpose. And he holds positions in both mechanical engineering and civil and environmental engineering, and he works at the interface of many fields, not just these two, but also you know, biomaterials and soft materials as well. And many of you know Professor Balogun. He doesn't need a long introduction. He is, of course, a fantastic lecturer, and he teaches the very popular mechanical vibrations course that many of our students in civil engineering take. And this talk will feature some of his recent work, I believe, with Professor George Wells in collaboration with his group, really at the interface of mechanics and environmental sciences, and focusing on the mechanics of biofilms and other related soft materials. So it's a really great pleasure to have him today to give our first seminar. You know, I'll let him take over. Before we start, though, we should probably figure out how we want to do questions. Shay, do you have any preference on how you want to handle questions? How do we?
Yeah, you can interrupt me in between. It's to have a more collaborative discussion. That's fine with me.
Okay, okay. If you like, you can type your questions in the chat box. I guess that's probably one less intrusive way to ask them.
Right.
But you can also, for quick clarifications, I guess speaking out would be also okay. So with that, let's get started.
Thank you, Sinan. Certainly, it's a pleasure to lead off the seminar series this quarter. I'm going to spend the next 40 plus minutes or so just discussing aspects of the work we do in the area of soft materials, particularly focusing on experimental approaches for characterizing viscoelastic properties. I think a good place to start is to sort of introduce myself, in particular focusing on the kinds of work I do. Sinan has already covered most of those, but I just wanted to show you a few pictures of some of the work that we do. So in general, we work with elastic waves as well as vibrations, and we're particularly interested in understanding and how these waves travel in various materials and at different length scales. So for example, at the atomic scales, we're interested in understanding how interfaces and defects in mixed dimensional semiconductor heterostructures, how they control the flow of phonons, or which are quantized lattice vibrations or lattice waves that control heat flow and then the intrinsic thermal properties of those materials. We're also interested in a mixed dimensionality. So for example, when you combine a 0D material like a fullerene with a 2D material like graphene or molybdenum disulfide, and there we're trying to understand how These interfaces control the flow of mechanical vibrations between the 0D and the 2D materials. These materials are designed for optoelectronic applications as well as nanoelectronic applications. So we do this work in collaboration with some colleagues at the Material Science Department, under the auspices of the Northwestern Materials Research Center. Um, we're also leveraging some of the ideas that we learned from atomic systems to replicate, say, the geometric properties in structural scales to create periodic structures and lattices that behave as metamaterials or phononic structures that ultimately can be tuned, for example, to serve as tunable mechanical filters or tunable mechanical diodes that allow you to control mechanical wave propagation in unprecedented ways. So we leverage things like bending, deformations, nonlinear coupling between mechanical elements as levers in order to control wave propagation in these materials. And then the topic of this talk today focuses on the biomechanics of soft materials. I will talk about biofilms, as well as later on in the talk, I will just introduce some recent work in the area of understanding the mechanics of glaucomatic tissue, focusing on wave propagation in the cornea. So that essentially captures the summary of what we do. So when you think about waves, when you think about mechanical vibrations at different scales, that's at the heart of the work that we do in my research group. Okay, so in this talk, I'm going to start off with a background and motivation for the work we're doing in the area of soft materials. I'll present some experimental approaches that we're developing and that we've explored over the last few years, some elastic wave modeling approaches that we've directly worked on in the context of viscoelastic wave propagation. And then I'll present two case studies, one focusing on environmental biofilms that sort of in this sort of plate configuration, and then granular biofilms that have some curved geometry associated with it. I'll present some conclusions, and then I'll present some ongoing work that we're doing in the area of understanding how waves travel in the cornea, and we're going to use that as sort of a diagnostic tool to diagnose the progression or development of glaucoma. Okay, so a good place to start from is to ask the question of what is a biofilm? So that's sort of the focus of the talk, the heart of the talk. What really is a biofilm? And so when we think about biofilms, we think about bacterial cells, right? So the cells can grow on surfaces. So when they want to colonize a surface, they typically secrete, a series of biopolymers, and these polymers includes extracellular DNA, proteins, and polysaccharides, and these proteins essentially encapsulate the bacterial cells, preventing them from chemical attack from the environment. Ultimately, these biofilms provide this dynamic architecture that encases these bacterial cells, and it provides protection from the environment, from chemical attack, for instance, like antimicrobial chemicals, but also it's the framework through nutrients from the environment are essentially sent through to the cells that aids in their growth and their development over time. So the biofilm itself is an important structural material. It consists mostly of 90% water, so it's in a gel form. What you will see as we go along is that the structure and mechanical properties are highly complicated, and so understanding that coupling is what the focus of this work is. Now, these biofilms are ubiquitous, so they grow everywhere. One example, when I explain the work I do to my kids, I always tell them, well, I'm interested in biofilms because they grow on your teeth, and that's why you have to brush it every day. And so ultimately, they tend to understand that aspect of the work. So they grow on surfaces, and they're ubiquitous. They grow on any surfaces that essentially promote, the growth of these biofilms. And then I'm going to call this planar biofilms, but also besides growing on surfaces, they could also granulate. So they could essentially form, biogranules, and then they float around in water. And you see that this granular biofilms are essentially what is used in wastewater treatment applications for cleaning up used water. And so if we look at biofilms, they can be characterized as having detrimental attributes, meaning that they're harmful. One way to look at these is that this biofilms themselves acts as contamination vectors. So for example, when they grow inside water pipes, and they break off, when these biofilms break off over time, they essentially contaminate the water and spread that bacterial load into the water. And so they tend to spread bacteria in this way. They're very harmful when you think about surgical equipments, and so that's why those surgical equipments are typically sterilized quite frequently to remove any sort of bacterial load on the surfaces. But these bacterial cells, the biofilms can be intentionally grown. They can be grown for beneficial attributes. So for example, they can be used, the biofilms themselves can feed on the bacterial load, for example, in the water, and so clean up used water for reuse in that way. They also can be used as underwater adhesives. I know Professor Sinan Kitten does work in that area using this biofilms as underwater adhesives. They also have some beneficial characteristics, particularly in the area of biofuels and bioremediation applications. So in a nutshell, they have both, beneficial attributes as well as detrimental attributes. Now, how did I get into this work? Well, in collaboration with Professor George Wells, also in the civil engineering department, what we're interested in is to try to understand this complex material, which we call the biofilm. It's highly heterogeneous. It consists of multiple, microbial populations. Some of them are single species, multiple species. They're highly heterogeneous spatially, and so their properties vary, both structure as well as mechanical properties as a function of space. The other attribute is that these are living materials, so they adapt to external stimuli. So they adapt and they change their properties. And so it's been quite challenging, to obtain any sort of consensus in the predicted properties in the literature. And so what we set out to do was to try to understand how the structure, the internal structure, its chemical functionalities, and also their mechanical properties, how they're coupled together. And so this requires a suite of tools to be able to achieve this goal. And so what I'm going to talk about is how some of the methods that we've explored over the last couple of years to address these questions. We're interested in not only the viscoelastic properties, we're interested in also the adhesive failure, as well as the cohesive failure. Now, this is sort of a preliminary, this is a long-term project, so I'm only going to present the viscoelastic aspect deformation of these materials. I would also add that one of the complexities here is the availability of the right kind of tools to characterize these properties. And so if I move over to the next slide, this sort of summarizes where the state of the art is today in terms of characterization of the mechanical properties of this biofilm. So at the very small scale, what is being used are, AFM techniques. So this is an atomic force microscope, which essentially is a diving board with a nanoscale tip that is typically functionalized, and you use that to essentially pull on the biofilms at the local level, right at the tip. And so you can study, for example, the tension behavior or the tensile behavior of individual components of the biofilm. Other techniques that have also been developed at this scale includes the optical tweezers, where you take light, you illuminate a bead, could be a polystyrene bead. And based on the optical gradient forces on the bead, you can essentially tug on the biofilm locally and characterize both its tensile modulus as well as its relaxation rates. This can also be done using magnetic tweezers instead of light. So you take a magnetic particles in a magnetic field. And so you can use that magnetic field as a local actuator to deform the biofilm locally, and then you study its dynamics over time. On the large scale, sort of a longer length scale, we have instrumented tensile testing that has also been used to characterize the load displacement behavior. And these methods can also be extended for creep testing as well. And also the famous rheology measurements as well that has also been applied. One of the challenges with the results from these methods is that they're completely different. And that's because of the high degree of heterogeneity of these biofilms. On the smaller length scales, you're essentially looking at sampled properties, and you really need large sample sizes in order to get representative average quantities. And then on the longer length scales, the measurements are essentially volume averaged over the intrinsic heterogeneity, so it masks the underlying heterogeneity in the materials. And so we came in mostly interested in looking at the properties within the mesoscale. And within the scale in particular, there are reports in the literature that suggest that the structural properties at the mesoscale, instead of this range between 100 microns and 1 millimeter, ultimately control several processes involved in the biofilm growth and proliferation on surfaces. So things like biofilm attachment and detachment from the surfaces, as well as the rates of mass transfer. And so that's sort of how we formulated this problem. So to be able to begin to address these questions, we wanted to know, can we develop a technique that ultimately can allow us to access structural properties as well as mechanical properties within this mesoscale length scale. And so to do this, we researched two different methods that I'm going to discuss today. One of which is the optical coherence tomography technique. This technique is essentially used in the literature for morphology characterization, both at the micro and the micro scales. So essentially, it consists of a light source, a low coherence light source that is directed into a sample. And that sample itself has to scatter the light. So it scatters the light within that volume, and that light is mixed with a reference beam, and ultimately that's used to create an optical interference, okay? And so it's basically a low coherence optical interferometer. The local scattering information is what is used to create the contrast within the material. And the source of that contrast is the index of refraction. So the index of refraction, you will see later, is it is directly correlated with the morphology of that partially transparent material. So some of the interesting attributes of this measurement technique is that it's non-destructive, it's rapid, it allows its own mechanical contact with the material. The resolutions and the micron scale, both in the lateral as well as the depth, the axial directions. So here's some initial work, results we obtained in a biofilm that was grown in the lab. So the picture on the right here is just an optical image from the side showing a fairly thick biofilm. And what we did was that we profiled the porosity distribution in this material based on the measurement of the light scattering images. So what you're seeing here is a two-dimensional image. That's the depth, and that's the lateral direction along X. And the heat map you see is a measure of the light scattering intensity. The dark spots are regions in which you have water-filled voids or channels, and then the hot spots there represent the actual polymeric substance, the extra polymeric substance, or the biomass, if you will. And so from images like this, both in 2 and 3D, one can analyze them and obtain areal porosity as a function of depth. And this would help us characterize the volume fraction, right, of the volume fraction of water within this biofilm. So that's one attribute that one can get from this. You can also characterize roughness. So for example, on the surface, you can characterize the roughness of the surface. And this is one major attribute of these biofilms. They're intrinsically rough and also highly heterogeneous, as shown in the picture. Okay. Now, um, having established this approach to characterize the morphology of these biofilms, again, what we're interested in is how do we characterize the viscoelastic properties. And so the way we're doing that within the optical coherence elastography technique is that we're leveraging the high displacement sensitivity of this OCT interferometer. Just to go back one slide, I'm going to use this acronym OCT to represent optical coherence tomography. And the OCT primarily gives us morphology information. What we did next was to introduce elastic waves into the sample and then used the high sensitivity of the optical interferometer to detect the local vibrations at different points within the biofilm. And so by tracking the propagation of elastic waves within the biofilm, we're able to characterize the wave speed. And then we couple that to an elastodynamic model to be able to back out the elastic properties. So that's the essential approach. So the way we do our measurements is we take a piezoelectric actuator, which is connected to a mechanical actuator, just a needle syringe that taps on the surface, right, at the frequency at which the piezoelectric actuator is driven, and that creates mechanical contact, periodic mechanical contact at the biofilm. And these are very light mechanical contacts. So they're not really large deformations, such that our displacements are in the linear elastic regime. And what we do is we record the displacements as a function of distance within this biofilm, and then we do some processing to determine the wave speeds. And I'll get more into great detail on how we do this. So this technique is called the optical coherence elastography. Elastography here essentially means we're recording this elastic wave propagation and using that as a means of backing out the viscoelastic properties of the biofilm. Okay, so the technique is non-destructive. Again, it's rapid, and it allows us, its single measurement lasts, about a second, a couple seconds or so. So it's relatively fast. And so that allows us to adapt to changing viscoelastic properties as these biofilms adapt to their local environment. And what we're measuring is the viscoelastic properties. And I'll talk a little bit later about the wavelength of these elastic waves. So the picture you're looking at on this, in this picture here represents a propagating elastic wave, and the heat map represents the intensity from this interferometer. Each point is the signal at each point represents the phase of the light, scattered intensity, which depends on a combination of factors: the outer plane displacement, local outer plane displacement of the sample, and the local index of refraction. That's essentially what determines the contrast at this point. So you see several stripes. The distance between the red stripes or the blue stripes represents the wavelength of the elastic wave. Okay, so by exciting at a certain frequency, you record the wavelength of the elastic wave. The combination, the product of the wavelength and the frequency gives you the speed at which that wave is traveling. So that's essentially how we use the measured data to obtain the elastic wave speed. So since we're measuring elastic wave propagation in these materials, I thought it would be a good idea to sort of just do a quick summary of what these elastic waves are. So elastic waves are essentially elastic energy that travels within a material through local displacements of the atoms about their equilibrium positions. Okay, so for example, in this case, let's say you have a baffle that is oscillated at a fixed frequency. These particles you see are just trackers that essentially oscillate about a fixed point. And as a result of that, you see this energy flux travel from the left to the right in the screen. So that's a compressional wave in which the propagation vector is along the horizontal direction and the particles are moving along the horizontal direction. We also have shear waves. Shear waves consist of waves with orthogonal polarization to the direction of wave propagation. So the waves are traveling from left to right and the particles are moving up and down within the material. And then also we have surface waves, and I'll talk quite a bit about the surface waves in this lecture. very similar to what you would have observed when you drop a pebble in a pool, where you see this ripple pattern form. Those are examples of surface waves. In solids, they're called Rayleigh waves, and they have elliptical polarizations, meaning that they both have both in-plane displacements and out-of-plane displacements, with the out-of-plane being dominant, and the amplitude varies with position and with frequency. The penetration depth of these waves is typically on the order of a wavelength. And so the distance between the humps in this picture represent the wavelength of the surface wave. So the same thing if you look at a water wave, or this wave created by dropping a pebble in water, all that disturbance is limited to a shallow depth from the surface. And so this is a sort of characteristic of these surface waves. So I want to talk about how we actually use these surface waves to quantify the viscoelastic properties of a soft material. Okay, so if we take an example of a semi-infinite elastic solid, and then we strike that surface at a fixed frequency, what we do is we create a variety of waves. So we create bulk elastic waves. So the fastest traveling wave is a compressional wave. And then that's trailed by the wavefronts of a shear wave. That's what you're seeing here. So the combination of the compressional wave and the shear wave are bulk waves, so they travel within the bulk. And then you also see a wave that is limited to the near surface region here, which is the surface wave, or the Rayleigh wave, okay? And this picture you're seeing here is just a finite element calculation of the wave modes that are excited in a semi-infinite elastic solid, by mechanical contact. Now, the speed at which these waves propagate, for example, the compressional wave CL, depends on the elastic properties of the solid and its density. Okay, so what you're seeing here are two properties, the first and second Lemay parameters, and rho here being the density. And so by measuring the wave speed and knowing the density, you can determine the Lemay parameters by simply measuring the speed for a compressional wave and then the wave speed of the shear wave. Okay, so two unknown parameters, two measurements, and then you can quantify it. However, when we deal with biofilms, it's not as simple as what I've described. The reason being that these biofilms are grown on substrates, so they have some finite thickness, and we have to take into account the boundary conditions. So for example, the biofilm on one hand is exposed to air on one side, it could be exposed to water, and also it's grown on different types of substrates. So they could be rigid or soft substrates. So that traction at those interfaces have to be taken into account. So typically what happens is, rather than see this bulk waves, these waves essentially bounce back and forth within these layers, and then they create what we call guided elastic waves. And the speed at which those waves travel depend on the geometric properties, things like the thickness, and also the elastic properties of the biofilm. So we have to take all these into account, and this increases the complexity of the problem. And so naturally what it lends itself to is doing some sort of numerical analysis. And I'll explain exactly how that's done. So we're going to set up an elastodynamic wave model, and then use that to predict the wave speeds in these materials as a function of frequency. And I'll explain exactly how that's done. Okay, so again, going back to the problem statement, so we have a biofilm on a rigid substrate. And so the substrate is essentially free of any deformations because it's much stiffer than the soft gel-like biofilm. And then we account for the boundary conditions of the surrounding media. So the first thing we do is to formulate the elastodynamic wave equation in terms of the Lemay parameters, lambda and mu, okay? And this is a partial differential equation which is formulated in terms of the particle displacement vector, so u. And lambda star represents the complex Lemay parameters, And that's formulated in terms of the Kevin Voigt model. So we make some assumptions that the biofilm itself is heavily hydrated, so consists of over 90% water. And so it acts as a soft gel. And we assume that the constitutive properties follows the Kevin Voigt model, and in which case we can define in the frequency domain first and second Lemay parameters in terms of frequency dependent real and imaginary components of the elastic properties, so lambda, the real part, and then a viscosity term in the complex part of this properties. Okay, so having defined this, we go ahead and apply the potential functions in order to simplify the analysis of the problem. So we use scalar and vector potentials to sort of decouple the compressional wave equation from the transverse wave equations. And we do this. This is called the Helmholtz decomposition. So if you've done some elastodynamic wave modeling, this is a fairly familiar approach that is used. And then we reformulate the stresses and displacements in terms of those potentials. And ultimately, we apply the boundary conditions. Okay, so the boundary conditions in this case would be traction-free surface, particularly if it's exposed to, the biofilm is exposed to air, rigid boundary condition at the bottom surfaces. And this is what is expressed using this in the vectorial form at the bottom. So when you apply the boundary condition, and I apologize for this, certainly the busy slide that I have here, what you end up with is a circular equation that ultimately the solution to that circular equation tells us of all the possible modes that can propagate in that biofilm. And so that's what we're going to look at next, okay? So let's look at the solutions to the circular equation. So let's assume, for example, that we have an elastic layer on a rigid substrate. So just a purely elastic layer on a rigid substrate, and then you have air, so it's traction-free at the top surface. When we solve that circular equation, we end up with what we call a dispersion relation. So several curves in this picture. What I want you to take home is that each point on this curve represents a plane wave, right? Represents a wave that travels at a certain speed for a corresponding frequency. Now you could have many, many modes in this material. So for example, this horizontal line represents a transverse wave, just a shear wave that propagates from left to right, leading to vertical, out of plane vertical displacements of each point in this elastic layer. The curved lines represent guided waves. So they represent standing waves in the thickness direction, but they have a propagation constant, right, in the horizontal direction. So they are standing waves that are traveling from left to right in this film. And they travel at different speeds depending on what their wavelengths are. Okay, the slowest order mode, which is a guided wave, asymptotes to a constant speed, and that speed represents the wave speed of a Rayleigh wave or a surface wave. So you can imagine at very high frequencies, the wave no longer interacts with the boundary, and so what you end up with is a surface or Rayleigh wave whose energy decays as a function of depth, and at the appropriate frequency is essentially decoupled from the rigid substrate. Okay, let's see what happens if we change the boundary condition. So we replace now the air with water, right? So now there's some coupling of the energy into the water, the elastic energy into the water. What you see now is the elastic wave dispersion curves, they change. One very clear change you observe is that the Rayleigh wave speed actually reduces from 2.8 meters per second to 2.5. And the reason is because of the presence of the water loading on the surface, right, creates a delay in the wave speed as these waves travel on the surface. So it's no longer a Rayleigh wave, but actually it's a coupled wave in which the energy of the elastic wave couples into the water and the elastic layer. The other features in the curves are essentially the same. You have the shear wave speed, which is this horizontal constant line, and then the other curves representing the guided wave, so the standing wave modes that travels from left to right. Okay, so now let's see what happens when we introduce a viscoelastic layer. So in the presence of a viscoelastic layer with water loading, the curves essentially change, keeping in mind that one reason why that happens is because now we have frequency dependent elastic properties because of the Kevin Voigt model that is applied. And so rather than have a constant transverse wave speed, we actually have a frequency dependent shear wave speed. So that's one unique distinction that results from having a viscoelastic layer. So now the wave speeds become frequency dependent. And similarly, this horizontal asymptote that we observed previously for both the water-loaded surface and the air-loaded surface also becomes frequency dependent. So that's one major distinction that we see. And then another key point to note is the thickness. Okay, so as I mentioned, we grow these biofilms in surfaces and then they have some finite thickness. And so if I change the thickness from 10 millimeters to 1 millimeter, that controls the number of modes that are excited within this elastic layer for an equivalent frequency bandwidth. In the dispersion curves. So so clearly, when you have thicker layers, one has to think about it, the number of wavelengths that you can wrap within that thickness. So that would change as the thickness changes. So that's one major distinction that you see with changing the thickness of those films. So in a nutshell, this sort of shows the sensitivity of this wave speed, okay, to both the elastic properties, the viscoelastic properties of the biofilm, as well as the geometric properties. So having established this understanding, the next thing we tried to do was to validate models in systems that are well recognized in the literature. So we took an agarose gel, Phantom. So this is just agarose gel. So it's a hydrogel that we made in the lab. Again, typically has a very high water content. And then this picture you're seeing here is the structural image of the gel. Again, with a water layer at the top and air. So it's very homogeneous. So you can see the structure image shows no features at all. So it's featureless. And now when we do our OCE measurements, so this is the stripes you see in this images represents the wavelength. So the distance between the red patches represents the wavelength of the elastic wave that we send through the sample. And then we can determine its wavelength and we know the frequency of the source. And that allows us to determine the wave speed. Okay, so that's the way this method is done. So we change the wave speed, we record the wavelength and then obtain our wave speeds. Right, so these are some measurements in this agarose gel for two different thicknesses, 1 millimeter and 10 millimeters. And for a 1% weight to volume ratio of this agarose gels. And so what you see here is for the 1 millimeter, curves are almost flat. However, for the thicker layer, you sort of see this increase in velocity with frequency. And if we increase the weight percentage of the agarose gel, what we see is an increase in the wave speed. So when you increase the weight percentage of the agarose gel, so you increase the amount of cross-linking in this polymers, and ultimately you stiffen it up and the wave speed increases. So that we see in this measurement. So these are all experimental measurements. And so the next thing we tried to do was to determine, using our model, we fit our model to the measured data, and then we sort of back out the viscoelastic properties. So again, we're focusing on the complex viscosity as well as the shear modulus. I must have mentioned this earlier, that because of the high water content, we're assuming incompressibility. So we can assume the Poisson's ratio is 0.5. Okay, so these are comparisons of our measurements, the wave speed versus frequency to our model. And from these measurements, we're able to extract the shear modulus and the complex viscosity. And we don't necessarily, we can't say, okay, these measurements are accurate, so we need to validate these measurements against the standard. And so what we did was to use the rheometry measurements. So in the rheometer, we're able to record both the shear modulus and the complex viscosity. One distinction between these two measurements techniques is the different time scales. So again, we're looking in our measurements where our actuators operate between 1 kilohertz and 4 kilohertz. However, in the rheometry measurements, the rates, the deformation rates are limited to less than a kilohertz, and so there's clearly some distinction there. But to the first order, the shear moduli tend to be comparable, and so what we see here are the estimates. I think I move forward a little bit. So the estimates of the shear moduli for both samples are in good agreement with the rheometry predictions. These measurements were made in a rheometer in the material science department. And so that gave us confidence about our predictions of the shear moduli. And ultimately, we can now apply these to the biofilms that we grow in the lab. Okay. So I'm going to move on to the next slide where we start to show some of the case studies in the biofilm. So this is an example of an environmental biofilm that is grown in the lab. So this was done through a collaboration with Professor George Wells. So his postdoc assisted us with growing these biofilms in a rotating annular reactor. So these are highly complex biofilms with multiple microbial populations. So it's called a mixed culture biofilm. And the picture you're seeing at the top here is the structural image. So you can see, as I mentioned earlier, the contrast you're seeing here is light scattering. So regions with dark contrast represents water-filled voids, right? And the wider regions are regions in which you have the biomass. So it's a nice soft gel-like material, highly heterogeneous and rough on the surface. When we make our OCE measurement, so this is an OCE measurement at 0.6 kilohertz. Okay, you can actually see the same contrast reflected within the OCE images, and the reason is because each pixel point is directly proportional to the outer plane displacement of the sample, in addition to the local index of refraction. So these two images are superimposed on each other. Okay. And the other thing we notice here is not only do we see the stripes, but also the intensity of those stripes decays as a function of distance due to viscous losses in this material. Okay. So now what we do next is we extract the wavelength from the stripes. We know the excitation frequency, and ultimately this is all linear elastic, so we can determine the wave speed as a product of the wavelength and the frequency. Okay, so that's what we've done here, and what you see is a wave speed that increases almost linearly with frequency. And if you recall from one of the pictures I showed in the modeling section, that the reason why this is happening, particularly in regions where the wavelength is actually much smaller than the thickness of the biofilm, which indeed is the case within this frequency range, one should expect in an elastic solid, one should expect a constant wave speed corresponding to the Rayleigh wave speed. But in fact, we see this dispersion behavior where the wave speed increases with frequency, and that's as a result of viscoelasticity. So this sort of confirmed to us that indeed these materials are viscoelastic, and we're able to fit two parameters to our dispersion curve, so the shear modulus and the complex viscosity, to be able to determine the viscoelastic properties of the material. So this was sort of the original work we did, and this work has been published in Soft Matter, I think last year. Okay, and something else to mention, I forgot to do that, is that these properties are certainly within the ballpark of what is expected for the extra polymer substance, which constitutes the hydrated biomass within a biofilm. And so that gave us a lot of confidence to move on, that we're sort of on the right track. The next example I'm going to show is a granular biofilm. So it's certainly geometrically more complicated than the planar biofilms grown by this rotating annular reactor. And the granular biofilms essentially granulate rather than, they're not attached to surfaces, they granulate to form the spherical balls. So this is what they look like. We obtained this from a wastewater treatment facility in Illinois. And typically they have diameters in the range of 3 to 5 millimeters. And what we were interested in doing is to understand exactly how the internal structure, how that plays a role on the viscoelastic properties. So the literature suggests that this granular biofilms have sort of a layered architecture. And so this granular biofilms is supposed to, this is what the literature suggests anyway, they're supposed to have a gradient in oxygen concentration from the surface all the way out to the internal region, right? And as a result of that, you can almost think about this biogranules as layered, layered spherical films, right, stacked on top of each other. And so this is sort of our working hypothesis going in what, based on what we gleaned from the literature. And we wanted to see if this approach can be applied to characterize sort of a layer dependence of the viscoelastic properties in this biogranules. But the first thing which we attempted to do was, without assuming this layering, we thought the best thing to do would be to essentially take a section of this biofilm as shown, and then sample its local wave speed as a function of position. So if indeed we have this representative layering, that should be reflected in the wave speed as a function of position. So that's sort of what we set out to do, to probe the local spatial variation in the elastic wave speeds, okay? I'll just give an example of how this is done. So it requires high spatial resolution to do this. And so here's a model experiment that we started out with. So we took an agarose gel and we fabricated this in the lab in such a way that we can vary the concentration of the agarose gels laterally from 1% to 1.5%. So this is a sort of 3-layer agarose gel. And the middle layer is 1.25%. Agarose gel weight to volume ratio. And what we expect in this, with this contrast in this, the concentration of these gels, is we expect to see an increase in the wave speed as we go from the left to the right. So that's sort of the expectation. Here is an image showing the measured data. So these are time-lapse images that we use to record the propagation of these elastic waves from the left to right. And one of the things you clearly notice here is that the wavelength, the distance between the hotspots essentially increases as you move from left to right, indicating that indeed, as you move from the 1% gel to the 1.5% gel, you see a corresponding increase in the wavelength or the wave speed of the elastic waves. So we do some processing, we convert this data into a time, arrival time versus lateral position image. We call this the acoustic B-scan. And the images you see shows this dotted line between the line representing the lines of constant phase, the slope representing the wave speed. So we're able to extract the wave speeds directly from these images in this way. So suffice it to say, we see a gradually increasing wave speed, almost constant on the left-hand side, right around 2 meters per second to about 3.5 meters per second on the right-hand side, and we go through a transition region in the middle. And so that really shows us the contrast between the 1.1% agarose gel and the 1.5% agarose gel. So this representative layering is what we see in the wave speed that we measure. We've done a lot of analysis to figure out what the spatial resolution of these measurements are. We're able to resolve this transition across this sort of abrupt change in the agarose concentration with a resolution of about 0.5 microns. So that's comparable to the wavelength of the elastic waves in this material. we can go to higher spatial resolutions. I think our limit is somewhere around 50 microns by increasing the frequency. So typically by working at high frequencies in the tens of kilohertz range, we can increase the sharpness of these transitions in the wave speed. And so I can discuss this later if you're interested. Okay, so having gone through all of that, now we can go back to the biogranule. So we sectioned the biogranule. So this is an optical image of what it looks like. And then we recorded the wave speed as a function of position along this granule. So here is the structural OCT image and showing that it's almost, there's no internal heterogeneity that is clearly identifiable or zoning, as it were, in the porosity. So that's at least what we can say from these images. And then when we analyzed our OCE image, our elasticity image, and obtained the acoustic B-scan, and from the slope of this transition points, the lines of constant phase, we obtained our wave speeds, and that's what we're profiling in this picture. And so what we were able to identify is that right at the center of this granular biofilm, we actually have a reduced wave speed. So in indicating that the shear modulus is actually lower at that location, or the density is lower at that location. And compared to the regions around it. The other thing we notice is right around the boundaries, this circle that you're seeing here, we also see reduced wave speeds. So the first thing we can report here is that indeed these biofilms are not as simple as the sort of layered, two-layered concentric spheres that was reported in the literature. And we're still doing a lot of analysis on other granular biofilms to be able to predict the depth-dependent profile of the shear modulus as a function of position. So this represents sort of the first report in the literature on this layered dependence of either the elastic wave speeds or the shear modulus. So our conclusions from these estimates is that especially varying elastic wave speeds suggest an inhomogeneity in the mechanical properties and the density. Okay. And that the inner core is softer than the surrounding regions. And I will sort of provide an explanation for why that possibly is the case. So the next thing we try to do, again, bearing in mind that we don't want to destroy the biofilm by sectioning it. Instead, what we would actually prefer to do is to essentially record mechanical waves on the granular biofilms without sectioning. So we developed a circumferential model which allows us to track waves that propagate along the perimeter of these biofilms, which is what you're seeing in the picture. Here, that's the OCT image and the OC image of some alginate hydrogels. We obtained the dispersion relation in these materials, fitted it to our models. And again, we can obtain from these measurements the shear moduli and the complex viscosity. And one parameter that we need to make these measurements is we need to know the geometric properties. We're able to fit a line profile to the curved boundaries to determine its radius of curvature. So we do that and we're able to predict these properties. So I'm certainly conscious of the time, so I'm just going to skip through a couple of slides and get to the summary slides. If we add some bacterial cells to this alginate biofilms and repeat the same measurements, what we observe is that for the alginate spheres without any bacterial cells, we tend to get higher shear moduli compared to the case with bacterial cells. So that might explain why the central portion of these biofilms are essentially softer, right, than the surrounding. So the large concentration of bacterial cells within the core of this granular biofilms may essentially be responsible for its lower shear moduli. And so that's sort of what we're reporting here. So I think this is as much as I have enough time to explain. I think I might have taking a longer time than what when I practiced the talk, but I hope you're able to get something about how these measurements are done and what sort of properties we're able to extract from these measurements. I will just conclude with a summary slide. Let's move down here. Excuse me. Of what it is that at least I've been able to discuss in this slide, in this talk, is that the OCE technique is promising for viscoelastic characterization of biofilms and hydrogels. We can get not only the structural properties, but we can get structural and viscoelastic properties co-registered. And our spatial resolution is ultimately limited by wavelength. And in order to extract these properties, we need an elastodynamic wave modeling approach, which is what we've spent the last couple of years developing, both for planar geometries as well as curved geometries. And this work was supported by several grants from the National Science Foundation, and also several people worked on these. I collaborated with, Professor George Wells, Professor Aaron Packman. I don't want to walk through my slides to get to those acknowledgement slides, but I want to thank all the graduate students that worked on this project with me. Thank you very much for your attention, and I think I'll stop here.
Thank you very much, Shay, for a fantastic presentation. We have time for a few questions, and I see Professor Jim Hambleton has a question in the chat box. I'll read it out loud for convenience. He thanks for the seminar, and then with respect to the application discussed in the introduction, what is of most interest? Instantaneous response, like elastic properties, long-term response, viscous properties, strength of the medium, biofilm, and/or strength of the interface.
Yeah, so what's of interest is both the instantaneous response, which we characterize in terms of the shear modulus. So these materials are incompressible. And so we're mostly focusing on the shear modulus, and that actually controls the deformation of these biofilms under flow conditions. It's a property that controls how this biofilm deforms. We're also interested in predicting detachment, and I didn't have enough time to discuss how we go about that, but we're actually using high amplitude stress waves to detach these biofilms from the surface, and then to characterize the interfacial stiffness as well as the interfacial traction at that interface. So all of these properties are important to determine what controls the detachment of these biofilms from the surfaces. Ultimately, this is important in order for these biofilms to recolonize other surfaces, right? They have to detach and then move through the fluid and then reattach at other points in the surface. So I would say both the elastic deformation to control how the film deforms under fluid flow conditions, which is dominated by the shear modulus, but also detachment properties as well, which is the adhesion strength. And I didn't have enough time to get there.
Great. Thank you, Shay. Any other questions? You can certainly type them in the chat box. You may also speak out. That should be okay too, since we are a reasonably small participant group. I see 35, which is not that small, but I think we should be able to manage.
Right.
Any questions from the audience, from students? I guess I have one quick question, especially on that second part on granular biofilms, which I didn't know too much about actually. So you mentioned that there's a gradient in the mechanical properties.
Yes.
And you mentioned that there's also a gradient in the cell density as you go from the core to the shell.
Yes.
Can you comment on how morphology of fibrous constituents inside these granular biofilms change? Is much known about how the structure of the matrix changes or its constituents as you go from inside to the outside?
So not much is reported in the literature on the actual orientation of the fibers as well as its density. So that's actually an important property. In our measurements, what we're looking at are measurements on the scales of tens of microns to 100 microns. And so ultimately what we're going to see is average properties over that volume.
Right.
Okay, and so we may not necessarily resolve the property variations due to, for example, the fiber networks. Now, what we can say is indeed, if the fiber networks are highly ordered, if they're oriented in certain directions, that will naturally lead to anisotropy. So far, there's no evidence, particularly at the length scale of our measurements, that this is indeed the case. So most of these materials are isotropic, and that might have to do with the fact that these fibers are randomly oriented to begin with. But I can imagine that, the tension, for example, in the fiber would ultimately determine its sort of tensile property. So along the direction of those fibers, it will be stiffer than in orthogonal directions, where the sort of matrix ultimately would determine those properties. So, for example, shear and all of that.
Yeah, the reason I asked was in a totally different context. In
Yes.
Other types of biofilms, some papers argue that the contribution of cells to mechanical properties is almost negligible, and what matters more is which fibers are there, how much cellulose is there, and things of that nature. So I wonder if it's different in different contexts of biofilms.
Yeah. So what I have seen is that it depends on what type of property you're talking about. So in particular, if you're interested in the longitudinal stiffness, right, sort of a P-wave stiffness, because these biofilms are over 90%, their volume fraction of water is fairly high, over 90%, and then what dominates its longitudinal response as deformation is actually the water, so the water content itself. However, if you're dealing with shear deformations, right, because the water itself does not support shear deformations, right, the biofilm itself, the constituents of the biofilms ultimately controls that. And so focusing on shear waves or shear deformations allows you to tease out the contributing role of those constituent materials.
Yeah, that makes sense. Thank you very much, Shay.
You're welcome.
Maybe we have time for another question. Okay, there's a question from Fab. Would you say that rather that we could just use global properties instead of local properties?
I would say so again, the goal of this project is to understand the coupling between the structure at a micro scale, the sort of tens of micron scale, with the mechanical properties on that scale. And so local in the context of tens of microns to a few hundred microns, I can say, well, if we'll be interested in defining the local properties on that scale. However, if you're looking at properties at a lower length scale, so maybe properties on the scale of the fiber networks, then we can't get those properties using this method. So you might want to look at more localized methods like the optical tweezers and then the AFM based approaches. So it all depends on what length scale you're interested in. So I would say global properties average over the fiber networks within the biopolymers. Have a local in the context of resolutions on the scales of tens of microns to about 100 microns.
Great. Any other questions from the audience? Okay, I think that's probably it. So thank you, Shay, for a wonderful seminar. It was great to see all this work and learn more about the experimental side of things. And this concludes our seminar for today. We'll continue on next week at the same time. Thanks for all our participants, and see you next time.
Thank you.
Thank you. Bye-bye.